Hi, my name is Ravisen Beemadoo. I am a student at the University of Mauritius. Today I'll present about network analysis of protein-protein interactions relevant in atherosclerosis and rheumatoid arthritis. So a brief introduction about the biology of the diseases. Atherosclerosis and rheumatoid arthritis are both chronic inflammatory diseases driven by a complex pathophysiology involving molecular interactions between several genes and gene products, also known as proteins. Studies have shown that patients suffering from rheumatoid arthritis develop cardiovascular disease complications, the main cause of death and of high incidence globally. This may be due to a common inflammatory mechanism. However, the link between both diseases is still poorly understood, both at network and molecular level. So the molecular mechanism underlying the disease pathophysiology is still poorly understood. This study aimed to carry out network analysis of genes associated with these two diseases to understand how they interact and if this can possibly explain the pathology. The focus here was on the common genes and the functional implications in the pathways that underlie the pathophysiology of both diseases. This was done by detection of functional modules. So modules include the group of clusters of interacting genes or proteins and genes represented by nodes and the interaction represented by the edges. The approach used was to select and filter data so as to construct an integrated database. From this integrated database, disease genes were retrieved using structured query language, the SQL query, and mapped to the protein-protein interaction data from BioGrid to construct protein interaction networks. Network analysis was then performed to identify the topological features of the genes involved in the networks. In order to define the pathways implicated in both diseases and their biological processes, we use the gene ontology for functional annotation. So this flowchart here gives an overview of the steps involved in the methodology. The first step is data processing. The files were downloaded and parsed to an integrated database. We use disease genes, namely dysgenetic Malakoff diseases and CBI, to retrieve the disease genes, which were mapped to their protein-protein interaction data from BioGrid. Cytoscape was then used to construct and visualize the protein interaction networks. The network analyzer plugin in Cytoscape was used to identify the highly connected nodes based on degree and betweenness centrality as topological parameters. The hub genes were then functionally annotated using David and Klugo. So here is the link for David functional annotation using the gene ontology. Our main findings was that 43 intergenes, which are both inflammation and disease genes, were retrieved from the integrated database, and David functional annotation resulted in 5 significant geoterms. The table shows the significant enrichment for inflammatory response, followed by extracellular regulated kinase cascade and the signaling pathways. Main findings point to 7 hub genes, also common inflammation genes such as the vascular cell adhesion molecule, cytokines, tumor necrosis factor, and transforming growth factor beta-1, and lipid metabolism, including genes such as prostaglandin and the peroxide synthase. The gene ontology revealed the function inflammatory response. So this is already known about these genes, but in addition, we found the regulation of lipid metabolism and signaling pathways. From network analysis, we identified 26 hub genes out of the 43 intergenes having a degree greater than 10. This protein-protein interaction illustrates the interaction of these 26 hub genes. As you can see from the table, shows the genes having highest degree at the top, the vascular cell adhesion molecule. Using the molecular complex detection code, we identified 3 modules, and these were functionally annotated, which resulted in 3 significant functional modules. The first module was involved in tumor necrosis factor signaling pathway. So as you can see, the significance of TNF in the network is represented by the size of the node. The bigger the size of the node corresponds to a higher degree. The second module is involved in interleukin-17 signaling pathway, and we have the transforming growth factor beta-1 and interleukin-17 receptor and the other associated genes. The third module involves interactions between co-stimulatory molecules as part of the regulation of T-cell proliferation and differentiation. Based on the David gene ontology functional annotation, Using the biological processes, we constructed protein interaction networks based on geoterms such as the inflammatory response and identified pathways such as rheumatoid arthritis. So these two tables show the topological parameters computed showing the hub genes transforming growth factor beta 1 and tumor necrosis factor being common in both inflammatory network and in rheumatoid arthritis. Indeed, in rheumatoid arthritis, the same genes hence were identified as common inflammation genes. This protein interaction network shows tumor necrosis factor signaling pathway with the nodes and interactions involved with the same inflammation genes, including the vascular cell adhesion molecule, the tumor necrosis factor, and other associated inflammation genes. In addition, we found the interleukin-17 signaling pathway in T-cell differentiation and proliferation. So a possible explanation could be a dysregulation in both lipid and T-cell differentiation could cause inflammation. In this study, the geoterms were not only involved in inflammatory response, but also the negative regulation of lipid metabolism and dysregulation of T-cell differentiation and proliferation that could lead to autoimmune responses. The significance of inflammatory response could be our main conclusion and the common pathological feature that links both rheumatoid arthritis and atherosclerosis. Hub genes identified could represent potential biomarkers. For example, the transforming growth factor beta 1 was previously reported to be a marker such as a single nucleotide polymorphism. So this could have an impact on drug discovery and targets. Indeed, the detection of interaction modules points to molecular mechanism that causes the disease. With this, I will end my presentation. Thank you.